Hey guys, video 3. I realized at the end of when I went to sort of end the video, that I never explained a use for agrose. So a use for agrose is if you've ever, um, in biology, heard about how we separate DNA, right? We use a agrose gel, okay, whenever we perform our electrophoresis. It's very good at sort of holding on to things for us. So again, we use it for its structural component nest, but it also allows water to pass through, which is really important, right? That's what's good about agrose. It allows water to move with it. So, and then we talk about these glycosaminos, these glycosaminoglycans, right? They're kind of hard to say, but glycosaminoglycans. And they're these repeating disaccharide units, right? There's 6 specific ones that are shown here. Some, like heparin, have a high negative component, which is why heparin is such a good anticoagulant, right? It can grab all those, it's sort of attracted to a lot of things, right? Lots of interactions with things with all those negatively charged groups. And then you have this compound, which is actually really important in sort of lubrication, right? So it works really well at lubrication. And you notice that for lubrication, we don't need a lot of negative, right? It only has one negative component. And so, but again, it's providing some amount of structural support, whether they're found in tendons or cartilage or in the extracellular matrix of the skin. I care less about you being able to know what's in what and more about, again, we're just driving home this idea of structural importance of these polysaccharide units. Another way in which polysaccharides are actually really important to us are in bacteria cell walls. Okay, so our bacteria cell walls are actually composed of either 1 or 2 bilayers and a peptidoglycan shell. Okay, and this peptidoglycan shell is made up of these 2 units as far as the polysaccharide chain is concerned, and it's a 1,4 linkage, and it is beta, right? It's a beta 1,4 linkage, and this guy, NAG, this is your N-acetylglucosamine, and then NAM, this is your N-acetylmuramic acid, okay? And so you have these sort of polysaccharide chains, then these polysaccharide chains are actually interconnected through these tetrapeptides, which are actually composed of amino acids, right? So an L amino acid, sort of a D glutamate, which is isoglutamate, L lysine, and then D alanine. And then depending upon how this L lysine links up, we know that this is either going to be gram-negative bacteria or gram-positive bacteria. But the peptidoglycan, what that is the strong protective peptide polysaccharide layer. Why do we call it a peptide polysaccharide layer? Because it's not just the polysaccharide chains, it's the linkages, these tetrapeptide linkages as well. So let's look at the difference between gram-positive and gram-negative bacteria cell walls, okay? So if something is gram-positive bacteria, then it only has one membrane phospholipid bilayer. So here's our phospholipid bilayer. And on top of that, so directly interacting with the outside world, is our peptidoglycan cell wall. Okay, and here are our sort of polysaccharide chains, right? And we see that these polysaccharide chains are linked together through these tetrapeptide chains. That's the little circles, are the tetrapeptide chains. We'll talk about this green line in a second, right? Because there's another linkage here as well in gram-positive bacteria, because this is occupying a very large space, right? It's being a protective coating. Now, what about gram-negative bacteria? Well, in gram-negative bacteria, the phospholipid bilayers are sort of like the pieces of bread, and the peptidoglycan is the sort of meat in the sandwich, right? And notice these suckers are a lot closer together, right? It's much more smashed. It's not as big. Um, and basically, so this peptidoglycan, we call the space that it's in is this periplasmic space, and it's attached to these outer membranes through hydrophobic interactions, right? But notice a lot closer interactions, Well, since the other one had larger interactions, there needs to be more ways that it can be attached together. So in gram-positive bacteria, you have these extra crosslinks, which are pentaglycine, that give you like an extra bridging mechanism. We don't really need this in the gram-negative because it's much more smashed together, so you've got a direct sort of connection. The amino acids just link up to the amino acid. So the L-lysine of one peptide directly connects to the D-alanine of another. You have a direct connection. But in gram-positive, since they're more separate, there's actually a pentaglycine crosslink that sort of fits in the middle. But it's always L-lysine connects to D-alanine, either directly in gram-negative or indirectly through a pentaglycine crosslink in gram-positive. Okay? And so let's talk about animals in general. So animals, right, they have a variety of polysaccharides. They make up, when you compare to like bacteria, animal surfaces, they just have more diversity, right? We have more diversity in our structure, in our sort of composition, in our function. And it's really due why we have so much more diversity to us than, say, bacteria cells, is it's because of all the glycoproteins on our surface and the proteoglycans that make up our extracellular matrix. We have more connective proteins that exist, and we have polysaccharide structures that can regulate cell recognition and different forms of interaction. So we have polysaccharides that can do more than just store and provide structural support. And that's really what separates us, is our polysaccharides. We make more use of them. And the uniqueness, right, of the information in these structures, it can be determined by the enzymes that actually synthesize these. So we have enzymes in our body that synthesize these polysaccharide units, and depending upon the enzyme that is doing the synthesis, the polysaccharide may be more unique or less unique, depending upon its role as well. If you just need it to be storing, then it doesn't really need to have that unique of a structure. But if you want it to actually perform a task, like regulating cell recognition, then it needs to be more unique, right? Okay, so next up we have glycoproteins and their function, okay? So polysaccharides, as we mentioned before, we also call them glycans, so that's why they're polyglycans. They consist of monosaccharides and derivatives, right? So we have homopolysaccharides that contain only one kind of monosaccharide, and we have heteropolysaccharides that contain more than one kind of monosaccharide, right? We have already talked about this. Now we want to talk about what glycoproteins are, right? Glycoproteins contain covalently linked oligosaccharide and polysaccharide groups, meaning they don't have to be necessarily these huge, ginormous chains. Sometimes they can be the smaller chains. They can be oligosaccharides, right? And we find these in structural proteins, enzymes, membrane receptors, transport proteins, immunoglobulins, all sorts of stuff. So what we're talking about here is we're talking about a protein that is attached covalently to some sugars, right? So, and as far as how this attachment occurs, we have N-linked saccharides and we have O-linked saccharides. And that is directly corresponding to how the sugar is attached to the protein, okay? How this carb or sugar is attached. So carbohydrate groups, right? They can be linked to polypeptide chains via the hydroxyl groups of serine, threonine, and hydroxylysine. Whenever it connects that way, it's going through a hydroxyl group, OH, so we call this an O-linked saccharide, okay? Or asparagine has an amide nitrogen in its side chain, and it can actually link through that amide group in the side chain of asparagine, and when it does that, that is referred to as an N-linked saccharide, okay? So these are the 4 amino acids we're looking at as far as how they attach to saccharides. Okay, so serine and threonine, which are 2 common ones, 2 of the common 20, and then hydroxylysine. This is a sort of uncommon amino acid. It's lysine that's been hydroxylated. Okay, and then asparagine, another common amino acid. So there's only one N link that we're going to talk about and 3 O links. Okay, and in this example, this connection is through an oxygen of serine. So this is an O-linked saccharide. Don't worry about the nomenclature and stuff. Again, I didn't require you guys to take organic chemistry before you came into this class. So there are some things that I cannot necessarily require of you because I don't expect you to have new information. Okay? All right. So let's talk about O-linked saccharides a little bit. So O-linked saccharides are often found in the cell surface of glycoproteins and in mucins. So think about what maybe a mucin is. Can you think of how we use mucin? Maybe think of a medicine with mucin in it, like Mucinex. All right, so we're talking about mucus, right? Large glycoproteins that coat and protect the mucous membranes in our respiratory and gastrointestinal tracts, right? The steric interactions between the sort of sugar, the carb portion of this, the saccharide portion, and the protein, they cause the peptide to fold in a very particular, rigid, extended structure. Okay, and this allows sort of these mucus molecules and stuff to form these really extensive intertwined networks at low concentrations. Because think about it, you don't want a lot of mucus in your respiratory system, right? If you have a lot of mucus in your respiratory system, what happens? You start coughing, right? It's too much mucus. A little kid, a little baby that's just born needs more mucus in their lungs because they do not have the strength to expand their lungs. So they need a thicker mucus lining to lower the surface tension so that it takes less energy for them to expand their lungs. We're not little kids, we're big grown-ups. So we don't need excessive amounts of mucus. So we need a way to sort of line without like just slopping it on, right? And these glycoproteins allow us to do this, okay? So they allow us to be sort of viscous and protect the surface from harmful environmental agents like anything that you're breathing in that you don't necessarily want to get to immediately get absorbed in the skin and interacting with other things. They allow protection, but at the same time, they're not drowning us, right? Because that would be a problem if it was too much mucus. All right. So these O-linked saccharides, they make these extended conformations to sort of lift functional domains of these proteins above the membrane surface. So you see that they can actually go straight through the plasma membrane. They can sort of go into the center and sort of chill out there, right? Or again, this is another example of one that's going all the way through. But these are your O-linked saccharides. So here's the protein, right? And your O-linked saccharides are here on the surface of this protein, right? So they can actually help separate the membrane from the functional domain, okay? So what do we mean? It means that it helps keep this functional domain, this sort of globular head, away from the surface of the membrane. So it adds a little bit of sort of structural integrity that helps keep this portion away from interacting with things that it doesn't want to interact with. It's almost like you've got this celebrity and here are your bodyguards, right? These little O-links are literally just like the bodyguards of your protein, and they keep everything from these unwanted interactions from taking place with other things that it may interact with. Okay, so what's an example of an O-linked saccharide? Well, antifreeze glycoproteins are found in the blood. They actually allow fish to live in icy waters without, like freezing because they're in freezing waters. So they can actually inhibit formation of ice by binding to the growth sites of ice crystals. So what they do is they act as a blocker, again, a bodyguard, preventing this unwanted interaction from taking place. In this case, the unwanted interaction is the formation of ice. All right, so that's it for this video. Stover out.